And I kind of, the more I read about it, the more I was like, wow, I didn't realize how much we knew about aging and how much potential there is to intervene in the process and to sort of implicate and treat potentially different age-associated diseases. And so that combined with my interest in science communication, which I sort of developed through blog writing, etc. during university, I then sort of joined together and created my YouTube channel where I further read the latest papers, try to break down the different concepts.
Hey there, I'm Luca Fusar-Bassini. I'm a PhD student in computational biology at EPFL in Switzerland, and you're listening to the Biotech Futurist. The Biotech Futurist aims to foster deep understanding and discussion about exciting hot topics in biotech, but I want to say from the beginning that it is by no means rigorous in teaching the subject. And for the sake of outreach, sometimes we need generalizations that, of course, simplify the reality of the science behind what we're discussing. But I can say that my guests and I do our best to be clear and to go in depth. You can imagine to be out with me and my expert guest for a friendly conversation to get a general understanding and more curiosity, having fun as much as I've had recording this podcast. This podcast has no sponsors and any reference is not meant to support any commercial activity. This podcast is a solo effort, so if you wish to support me, I'd be grateful if you followed the Biotech Futurist on Spotify, Apple Podcast, YouTube, Instagram or your top podcasting platform and share it with your friends. With that said, I am excited to move on to today's conversation at the Biotech Futurist. Today I am lucky to meet Eleanor Sheekey. Eleanor is a PhD student at the Cancer Research UK Cambridge Institute, where she focuses on p53 biology. Eleanor has a beautiful YouTube channel where she distills the most innovative research in aging with a creative and engaging rhythm. I really encourage you to visit her channel and I'll put a link in the description. Watching her videos, I thought she'd be a great person for a review on the state of the art of aging research. So this episode will be more of an introduction than a detailed analysis of a single recent work. Eleanor, thanks for being here virtually with me. Can you introduce us to your story and what led you to outreach and aging?
Sure thing. So firstly, thank you for that really kind introduction. And yes, as you said, I'm currently a PhD student here at the University of Cambridge, where our lab studies cellular senescence, which I'm sure we might come to later, and how p53 plays a role in this process. But my introduction to aging, so to speak, probably came during my undergrad and master's. So when I was going to lectures, and we had a whole series of discussions on different processes such as neurodegeneration, cancer, stem cells, and how that all sort of was interrelated through the different hallmarks of aging. So that was my initial introduction to the field. And part of that involved writing essays, reading review articles, research papers. And I kind of, the more I read about it, the more I was like, wow, I didn't realize how much we knew about aging and how much potential there is to intervene in the process and to sort of implicate and treat potentially different age-associated diseases. And so that combined with my interest in science communication, which I sort of developed through blog writing, etc. during university, I then sort of joined together and created my YouTube channel where I further read the latest papers, try to break down the different concepts and just see what there is in the space at the moment and what the future potential is.
That's wonderful. So I think we'd be happy to start with an introduction to general concepts in aging research. Eleanor, can you describe for us what chronological, biological, and psychological age are and how the three of them relate to one another?
Yeah, so I mean, that's a good question. So obviously, I think a lot of people are comfortable and familiar with chronological age, which is just the time that's passed since you were born. So that's pretty easy for people to work out. But in terms of understanding the aging process, and so I should say that there is still no consensus over some sort of definition of what aging is. But what has become apparent is that if we want to be able to understand how to intervene in aging, we sort of need some metric to understand how these different interventions might be actually affecting someone's age. And so I probably haven't explained that too well, but essentially if you want to do like clinical trials for these different interventions, we can't just wait for people to die, and also there's no like easy comparison. You need like there's so many different confounding variables that we need some sort of biomarker of aging instead. And so that is where this idea of biological age was introduced instead, whereby you might have the same chronological age as someone, but you might be biologically younger or older. And that could be, if you were older biologically, might suggest that you're at an increased risk for different diseases, or your risk of mortality is higher. And so depending on the different clocks or different measurements that are used to define what biological age is, the risk factors might vary. And there's still a lot of controversy is not quite the right word, but there's lots of different ways in which you can do these measurements. And so what exactly biological age is telling us is a bit uncertain at the moment. But the idea is that it's a better proxy for your disease risk and a better measurement of your health than just judging your health on your chronological age. So being very arbitrary and saying everyone over 65 should do this or not do this. Instead, you could use someone's biological age and make a better prediction or a better suggestion of what someone should do to improve their health. And then you also mentioned psychological aging. It's been a while since I really read that, but the idea is that there's some evidence that if you think that you're younger than you are, you're more likely to also be healthier. And so I think, again, that's really interesting research. I don't know too much about it, but it's something that I think as we get better tools to understand the brain and how what we think and our sort of the things that kind of come along as pseudoscience at the moment, as we get better ways of being able to understand what it means, we might be able to infer some biology from it. But yeah, that is about as much as my understanding at the moment. So yeah.
Thank you. That's really interesting. And something that I learned from one of your videos is that there are oscillations in biological age. What does that mean? Yeah,
so oscillations, so there's many different ways in which you could define what an oscillation is. So there's one definition of oscillation. I mean, there's different time frames in which the oscillations could occur. So that also somewhat depends on the different methods in which you might measure someone's biological age. So the current main way that people refer to when they talk about biological age are so-called epigenetic aging clocks. And so these refer to DNA methylation marks that are present or absent on different locations in the genome. And you can use the presence or absence in a range of different locations and use linear regression to then approximate what someone's biological age is. But then you could also use different proteins present in the blood as a different biomarker. You can use things like facial skin images of the face. And I'm not entirely sure how those clocks work, but you can also use that to infer someone's age. And so some of these things like the proteins present in your blood may be more variable on a sort of day-to-day basis as compared to your face, which might be a more slower change. Honestly, I don't know. This is me being a bit suggestive. And so one thing that does change every day, or at least in a 24-hour time frame, is our circadian rhythm. And so there are certain physiological processes that happen on around a 24-hour timescale, and that's all sort of regulated through this core circadian clock. And so depending on whether you took one of these aging measurements in the morning versus the evening, you might actually see some slight variation. And then, so that's like a 24-hour timescale. Then you could also think about a monthly timescale, maybe 1 month, I'm feeling a bit ill, I've got the flu. That could definitely change some of these biomarkers as opposed to a month later where I'm feeling much more healthier or I've had more sleep. And so I think individually there'll be a lot of variation, but I guess if you look at the grander picture and you were more consistent and did it like every month, you might see, there's going to be some noise, but you might be able to see a sort of trajectory, which is what they've been able to at least somewhat infer from these population studies where they've initially derived these different biological aging clocks.
Nice. Guys, I think that we had bad luck not to record the video because she was really showing with her hands how these biological oscillations happen. So we had bad luck. Anyway, I also want to ask, what does it mean that aging is asynchronous?
So again, going back to what actually is aging, in terms of my definition, which for certain is not like the general consensus, I don't think there is any agreed definition of what aging is. But to me, at least, I see aging as being loss of homeostasis. So the way in which I mean by that is that obviously our different organs and our bodies are all in communication with each other. And so if we have some sort of insult, if we have some sort of viral infection, our body can deal with that and knows how to respond such that it then goes back to the original set point. If we get too cold, our body has physiological responses to warm us back up again, and vice versa. And so the way I see aging is that we sort of lose this ability to regulate the body. So there seems to be dysfunction in different tissues over time. And so when you say things like, is aging asynchronous, at least my understanding of that question is that different organs might age at different rates depending on different people. So there are certain factors that might cause one organ to age more than someone else. So smoking, you're more likely to see potentially a higher biological age in your lung compared to a non-smoker. And that's just so with skin, if someone was had a lot of UV exposure, you might expect them to have a higher biological age in their skin than someone who didn't, who wore a lot of sun cream. So I think that's kind of my understanding. And the interesting questions are, is there maybe some sort of temporal order that if your liver started aging, would that communicate with other organs and cause them to aging? And so obviously our body communicates through the bloodstream as one mechanism. And so if there was inflammatory factors being secreted from one organ, could that induce damage elsewhere in the body or sort of cause different effects? And so one way that could be is through the presence of senescent cells, for example. But this is kind of emerging research. I have heard that there is someone developing a body organ biological clock. So going back to these clocks we just been mentioning, the idea is I think they were looking at different organs within a mouse model or something. And so that might give us an actual understanding of how asynchronous aging is. And then also, how does it differ for different people? Is there how much of a genetic component is there to aging? Are some people more inclined to age in different tissues more than others? And besides it just being an interesting thing to look at, it's also interesting for thinking about interventions. If there are, because we can't define what aging is, and there seems to be different causes of aging, the treatments for different factors will therefore, logically, it feels like it would differ as well. So the more we can understand about this question and how heterogeneous it is, the better we can then know how to treat and intervene.
Yeah, that makes absolutely sense. So my next question becomes, when does aging begin? I mean, we have defined that aging is not a single thing, but it's many, many different things happening at once and in parallel with maybe different timescales. But at the same time, can one define when aging begins, and what is the concept of ground zero of aging, and potentially can we reduce and how this ground zero of aging, at least in theory?
Yeah, so I mean, that's also a really interesting question. And again, it's a little bit controversial. Different people have different ideas about when aging begins. And so some people might say, okay, as soon as you turn 25, as a rough estimate, as soon as you enter adulthood, that's when you start to sort of begin to age. And whereas the earlier period of time is development, and that we shouldn't sort of confuse development with aging. But then to go to your second part of the question about the ground zero stage, that refers to an initial hypothesis from Professor Vadim Gladyshev, who's at Harvard Medical School, who then actually has now published a paper showing some evidence for when this ground zero potentially could be. So what is this ground zero? So this is the idea that there's, at some point in our lives, the lowest minimum point our age could possibly be. So we're talking about biological age and how that is a proxy for chronological age. And so obviously we start at age zero chronologically, and however age we reach before we die varies. But the idea is that's a sort of one timescale. But a second timescale is our biological age. And so that doesn't necessarily have to begin at zero. And so we all start life as a gamete. We have an egg and sperm that come together and form the embryo. And so obviously they've derived from our parents. And one could speculate that the age of the sperm and the egg at the time of fertilization wouldn't be young. There has to be some sort of early rejuvenation event that enables the new embryo to begin from a biological age of zero. So there is this natural rejuvenation process that seems to happen each generation. And so what this more recent paper showed was by using one of these measurements of biological age, so DNA methylation, they could see in mice take samples from the very early stages of development, and they actually saw a decline in the biological age, which was really interesting, suggesting that there does seem to be this very early rejuvenation event at the very early stages of embryogenesis. And so it's not entirely sure exactly how that would translate to humans, but at some point in very early development, we seem to reach this biological age of 0, or whatever 0 really means in this context. And from then onwards, any sort of damage that could be accumulated over time, or whatever these different causes of aging actually are, we then see an increase in the biological age. So you could argue that, therefore, we begin aging after embryogenesis at that very early stage. But that also feels like a very kind of wrong thing to say, because obviously, as we've discussed, we're also going through development. And so, if we talk about anti-aging or reversing aging, we don't want to go back to that state. We want to sort of stay in the adulthood. So it's a little bit of semantics, I think, personally. But that is generally the kind of one biological estimate of when aging does begin. Again, we could criticize how much these different biological clocks reflect the aging process. Maybe DNA methylation is not the best thing to be using at early stages. Who knows? I think it's all kind of exciting but emerging research at the moment. So yeah, hopefully that answers your question.
Sure, thank you for sharing with us. So now I think we can shift to the cellular and physiological connotates of aging. I know you're a big fan of senescence, so my first question about cellular things in aging will be, what is senescence? How does it play a role in aging? What is SASP? And possible strategies to address this therapeutically, such as senolytic CAR T cells. And also, if you can describe briefly for us, what is the concept of vaccines for aging?
Mm-hmm. Yeah, so senescence is, I guess, what my entire lab studies at the moment. And so many people think of senescent cells, or they hear them be described as like zombie cells, which I always think that's a really bad sort of description of what a senescent cell is. So there are kind of two to three sort of main features of senescent cells. The first feature is that they enter a cell cycle arrest. So the cells literally stop proliferating. And they're not dead, which is what I guess sort of zombie, sort of maybe that's where the idea came from. But these cells are actually very metabolically active, and they're secreting a variety of different things into their environment, which is referred to as the senescence associated secretory phenotype, or the SASP, the SASP that we like to call it. And it's surviving. So the cell's very much not dead, it's just existing, it's just there. And so we think the secretory phenotype is important. And the reason we think it's important is because it comes back to where did these senescent cells come from in the first place and why they're existing. And so there's many ways you can make a cell become senescent. And all of them sort of revolve around some sort of cell stress. And so there could be DNA damage, oxidative damage. It could be a mutation in an oncogene that causes the cell to over-replicate. And effectively, all of these different mechanisms induce some sort of DNA damage response that activates the cell to enter a cell cycle arrest, some of which that could be mediated through one protein called p53. And so the cell stops proliferating initially to try and repair the damage, to deal with the problem. But if that damage persists, then the cell somehow, which is, again, we're not entirely sure how this decision process is made, but it enters into cellular senescence, where it then develops this secretory profile. And so coming back to the secretory profile, a lot of these different components include things like cytokines and inflammatory factors, and these are all sort of secreted into the surrounding environment. And the current, so there's a couple of different functionalities of this response. So in terms of the cytokines, they could bring in different immune cells to the area. And so in the case of some sort of DNA damage in cells, it could be important for a sort of wound response. So if there's some damage to the body, you accumulate senescent cells, they secrete things into the environment, that brings in some immune cells to then get rid of the damaged cells and get rid of them. And other things that they secrete are growth factors. And so if you got damage and you're going to get rid of those cells, you then need other cells to sort of replicate and replace the damaged cells. So all of these things that the senescent cells do, they seem to be beneficial. It's a physiological response. It's helping with wound healing. And in fact, studies have shown that if you add senolytics, so drugs that remove senescent cells during wound healing, you actually sort of slow down the process. So senescent cells are good in that context. But in the context of aging, what seems to happen is that if these senescent cells aren't cleared and they sort of just persist, that's one of their third attribute is their cell survival. They're just existing. And if they're just existing, they keep on chronically secreting all these different factors. And we think that sort of drives us in inflammaging. It sort of causes damage elsewhere. And it's just being a bit of a pest, really. It's just not doing anything. It's just not functioning as the cell should be. It's just taking up space, doing naughty things. And so that's why we think senescent cells could be also contributing to aging. So it's got good sides and bad sides. And so you've mentioned a couple of different senolytic strategies for me to talk about. So describe firstly what is sort of meant by a senolytic strategy. Effectively, it's a strategy that selectively kills senescent cells without affecting healthy cells. And so one way I sort of mentioned already is that you can use different small molecules that have been shown in vitro and to some extent in vivo, and so in mouse models, that they do clear senescent cells. But there's a whole range of different strategies that are now being investigated to see if we can understand the biology of a senescent cell and target its vulnerabilities to also clear them. And so you mentioned senolytic CAR T cells. I mean, I can speak about 5 minutes on CAR T cells to really explain how they work, but effectively they're sort of modified T cells, so T cells being part of the adaptive immune response. And so you modify these T cells so that they recognize receptors on the membrane of senescent cells. So the idea is they bind to senescent cells and recognize them, as opposed to the healthy neighboring cells that don't express this membrane protein receptor. And so if you can do that with the T cells can be T killer T cells, so they can actually then kill the senescent cells, or bring in other macrophages or other immune cells that can do that instead. I'm not an immunologist, so I'm sorry if I got that a little bit wrong. And then the more recent strategy is kind of similar but a bit different, is to also use these antibody drug conjugates. So again, exploiting the fact that senescent cells may have a different membrane protein profile compared to healthy cells. So antibodies recognize antigens. And so if you have an antibody that recognizes these membrane proteins that are present on senescent cells, but not healthy cells, you can then fuse to that antibody a sort of cytotoxic drug. And so if you deliver that into the tissue, you get the antibody recognize the senescent cell with the cytotoxic drug. It gets sort of endocytosed, so it enters into the cell, and then that payload is then delivered to then kill the senescent cell. I think, was that all of the things you asked me?
Yeah, that's really nice. Thank you. Now I'm opening kind of a very big topic where we should spend like 10 hours, but we will only spend like 5 to 10 minutes, which is protein aggregation in aging, and also the compelling but still very recent and explorative theory of contagion of aging happening between cells, how it can happen locally and more globally, and the highly discussed relationship between aging and neurodegenerative diseases.
Sure. Yeah, no, you're definitely right. Protein aggregation, definitely is a big topic. So, if we come back to like the different hallmarks of aging, so the hallmarks of aging is kind of a good introduction to understanding the aging field. There is some criticism over how good it is as being a paradigm for understanding the aging process, but in terms of just an introduction as to what different areas people are studying, it is a good way of thinking about it. And one of them includes protein dysfunction. So sort of coming back to sort of homeostasis, it includes things like protein misfolding, protein aggregation, and an increase in the sort of like errors that occur during translation. So that's when you go from like mRNA to the protein. And so obviously proteins are kind of like, as much as, you know, we love DNA, we love RNA and everything else that goes on in the cell, it's the proteins that possess all of the enzymatic activity that sort of do most of the chemical reactions that occur within the cell and within the body. And so having misfolded proteins means they're not in the correct conformation for them to perform the function that they should. Likewise with aggregates, if the protein misfolds, it might expose like hydrophobic surfaces that within a cell shouldn't, like, it doesn't, causes it to sort of prefer to bind to other proteins as opposed to staying soluble, and sort of causes these massive aggregates. And that's commonly seen in, like, some proteins are more prone to this than others. And one protein that's particularly prone to this is amyloid beta. And so one of the proteins that's particularly prone to aggregation is amyloid beta. And so amyloid beta, like aggregates of amyloid beta, are commonly seen in the brains of patients with Alzheimer's disease. And so you can see them inside the cells of patients with Alzheimer's disease. And another protein that also aggregates is a protein called tau. And the combination of these two protein aggregates have been attributed to the progression of Alzheimer's disease. But the interesting question is, are they causal to the process, or are they just a reflection of the aging process that happens in the brain? And it's still somewhat controversial. There's new evidence comes out all the time that either disproves or proves the hypothesis. And so I can't really say too much more on that, because as much as it was something I was following a lot, I'm definitely not up with the latest research in terms of understanding neurodegenerative diseases. But yeah, to come back in general about understanding what are the causes of aging, definitely anything that results in these dysfunctional aggregates from occurring is effectively taking your functional proteins away from doing what they should be doing. And so again, coming back to what I currently think aging is some sort of loss of homeostasis, a loss of dysfunction. It kind of makes sense that if you've got these aggregates, then you've basically disrupted the homeostatic mechanism within a cell. And so therefore, there are some approaches, at least with Alzheimer's that I know of, that are trying to develop small molecules or peptides that can sort of disrupt these aggregates, or to boost the levels of proteins that fold proteins correctly. So ironically, there are proteins that are involved in causing proteins to fold correctly. And so if there's ways that we can boost the proteostasis, so the regulation of protein folding, protein aggregates within a cell, that could be one potential anti-aging strategy. But unfortunately, I still feel like it is somewhat underexplored. Hopefully we'll improve upon that, but yeah.
Sure. So my next big topic to open is quite a big classic, which is telomere shortening in aging. So what is telomerase? How does telomere shortening happen? And what are the restoration strategies being proposed these days, such as small molecules, genetic interventions, or TERK stabilization? And what are the risks of such interventions?
Mm-hmm. Yeah, so for those who don't know, so telomeres are effectively long sequences that are found at the ends of our DNA. So our DNA is linear, so it has ends, and it's sort of organized into different chromosomes. So you might be familiar with the sort of X shapes of different chromosomes, but they're linear, so they have ends. And so that they have at the ends of them different telomeres. And so what are the function of these telomeres? Well, one function is to sort of act as sort of like shoe caps on shoelaces, the sort of protective ends to stop the DNA from unraveling. However, every time a cell replicates during DNA replication, the DNA replication isn't 100%. There's something known as the end replication problem. That means that on the ends of DNA, there's a gradual shortening of the base pairs each time the DNA is replicated. And so having the telomeres at the ends means that the telomeres are getting shorter every time the cell replicates. And so there's an enzyme called telomerase that is expressed in some cells, mainly stem cells, that can add extra repeats of these telomeres to the ends. And so that prevents the decline. But in many cells, telomerase isn't expressed, or it's expressed at very low levels, such that these telomeres are just shortening over time as the cell replicates. And so coming back to senescence, Leonard Hayflick first proposed the term senescence after growing some cells in a culture dish. And he noticed that after around 40 to 50 replications, the cells stopped dividing. They'd enter senescence. And now it's referred to as replicative senescence, that after the telomeres have shortened so many times, you've exposed the DNA end, and that's another sort of cell stress that is kind of dangerous for the cell to replicate when it's come to this position, because now that the DNA is vulnerable, it could get mutated or like aberrantly fuse ends to different parts of chromosomes and just basically cause mayhem within the cell. So the cell doesn't replicate and it enters senescence. And so therefore one might speculate that if the cell stops dividing after around 40 to 50 divisions, what if we could boost the activity of telomerase? What if we could add telomerase back to these cells? Would they continue to replicate longer than we usually expect? And so, of course, if you add telomerase to cells in a culture dish, they do, of course, replicate longer before they enter replicative senescence. The interesting question then is, can we apply this in vivo, so to different model organisms? And they've done that in mice. And I mean, it's been done many different times in sort of slightly different ways, where you can just basically introduce extra gene copies of the telomerase protein into the mice. And sometimes, so in some cases, you see increased cancer risk. And so if you think again about what telomerase is doing, it's increasing the lengths of these telomeres, which enables the cell to replicate more times. And so for a cancer cell, giving the cell telomerase means that they can just continue to replicate without having to worry about these aberrant chromosome connections that could occur. On the flip side, if you combine things like telomerase with other proteins such as p53, which is a tumor suppressor.
Random choice.
Of course, which is a tumor suppressor and responds to different cell stressors, then actually in combination it might be effective that you increase telomerase, which enables the cells to replicate more, but you're also increasing these sort of anti-cancer genes. And so there might be like a way that we can combine different genetic approaches together to actually improve the health span and or lifespan of these different model organisms. So again, how would you apply that to humans? That I know that there is some people who've actually injected themselves with telomerase gene. I'm not entirely sure how they did it. I don't know if this is also public knowledge. I think it is. I'm pretty sure it is. So obviously people are working on different gene therapies, ways to deliver genes into the body. In fact, if you think about the different mRNA vaccines we have for COVID, that's just one mechanism in which you can get information into a cell. In that case, it was an RNA copy, which in many ways could be something that you might want, because obviously with RNA, it's a sort of temporary expression of a gene. So maybe if we can work out a safe strategy, it might be to deliver telomerase to different tissues as an RNA vaccine. So you have a short-term exposure to the telomerase gene activity. You might be able to extend the telomeres. Obviously, the mRNA gets degraded, the protein essentially gets degraded, but maybe you've increased the number of times that cell could replicate, and maybe that could be beneficial for certain tissues that are going through a lot of replications, like skin or intestine. I haven't actually really looked in to see if anyone's actually doing that. It sounds like a good idea in my head now I've said it. And then there are potentially small molecules that could also bind to telomerase and increase its activity. I forget entirely which ones they are. There's also some ways in which you could increase the stability at the mRNA level to increase the expression of telomerase. So definitely, I think there's potential. I think it's critical that we work out a way that's going to be safe. So maybe that is either having a short exposure to telomerase activity or by combining it with different anti-cancer tumor suppressor genes.
Wonderful. So what about instead the genetics of aging? And also another very introductory topic I'd be happy to briefly explore with you is what about gut microbiome and aging? 2 questions in one.
Sure. Yeah. So talk about the first one. So there is one theory known as like the somatic mutation theory of aging, that as we age, we all get exposed to different UV radiation from different toxins, either through foods or through smoke, etc. That basically causes damage to the body. And, our cells get DNA breaks every single day, but our cells are good at responding to the damage and repairing it. But obviously, there's always going to be some breaks that sort of slip through. And over time, it's thought that we could accumulate these mutations. If the mutations are in important genes, the genes then get mutated, they lose their function. Again, kind of similar to all these other theories with like the protein aggregates. It's like some sort of loss of function occurs within the cell. And if it's in one cell, happens in multiple cells, you lose the function in the organ, you then lose systemic bodily function. And so there's been, with all the amazing advances in DNA sequencing, you can now actually look at different tissues and see the rate of mutations if you take tissues from model organisms at different time points. And so there's a kind of landmark paper that came out in Nature earlier this year that basically did this. I think it was in the intestine, and they did it in different model organisms where they had tissue samples from different stages of their lifespan. And what they found was that the somatic mutation rate inversely correlated with lifespan. Effectively, the higher the accumulation of these somatic mutations, the shorter the lifespan. And so that was actually quite strong evidence for this DNA mutation theory of aging. Obviously, it was just in one tissue. It was only a limited number of samples, but it does actually provide some rationale that DNA mutations could be one of these causal factors for aging. So I think we'll have to wait to hear more information on that story, but it's also one that I like to I like to think about because my research focuses on P53, as you already mentioned, and mutations that happen in this protein. And also I believe that it causes some sort of dysfunction to the cell. So kind of in line with something I think about a lot. But something I also think about a lot, but I don't directly work on, is the microbiome. So you mentioned the gut microbiome, which is, in our guts, but we also have microbiomes in our skin and also our oral cavity. And so effectively, we think our bodies are made of human cells, but actually there's a whole host of different microbes that are also residing within and on us. So that could be bacteria, viruses, fungi, and they you might think, oh, that's really bad, but they're really important for how our bodies function. So they're important for digesting our food. There's a lot of food like fiber and stuff that our bodies just don't know what to do with, but these different bacteria can break it down and produce like short chain fatty acids that are important for different things within the cell. I can't quite remember. But the bacteria themselves are also generating different factors that they can secrete into the environment. And so it's now known that the bacteria are important for our immune response and potentially also communicating with our brain. And so there's this whole gut brain immune axis, the gut axis that's being explored. And so there's a lot of really interesting research being done. The microbiome composition has also been shown to change with age. So going back to these aging clocks I mentioned at the start, they've done it with DNA methylation, with different protein changes, but they've also did try to create one by looking at how the gut microbiome changes with age, which is really quite interesting. And yeah, I think it'd be really interesting to find ways either to tweak the composition or to at least understand the sort of biochemical molecules that these bacteria are generating and understand which ones are beneficial and seem to be contributing to our health. I think there's a lot of things we don't understand about our gut microbiome at the moment. And actually, there was a recent paper that came out in Nature Aging last week that also showed that our, I think it was the skin microbiome, was also something that was super interesting. And because obviously our skin's exposed to all of the things around us, and it was, I only read it like the other day, but it was basically showing, if you looked at the composition of the skin microbiome, you could see even more distinct changes with age than maybe just looking at the gut microbiome. And obviously the skin's more accessible, so that could be another good biomarker for looking at biological age. So it's something I'm always looking into, super interesting stuff. But yeah, we're not just made of human cells, we also have to take care of our microbiome as well.
Yeah, I'm looking forward to all of your videos in the next few years dealing with microbiome at different levels in aging.
There's so much to talk about, yeah.
Yeah, maybe the next episode on this podcast with Eleanor will be entirely about the gut microbiome in aging. That would be nice. Anyway, I think we are short of time, so maybe we should jump to animal models and the central nervous system briefly. So one interesting case that you brought in your videos is the cloning of Dolly. Most of you are probably familiar with the case with Dolly, the sheep that got cloned as first mammal to get cloned, and she was quite a normal sheep for many reasons, and she had children, let's say. So, Eleanor, how does the case with Dolly relate to aging? Why it matters in aging research?
Yeah, so the case of Dolly was like one of the sort of most remarkable studies that you still learn about as you go through undergrad. And essentially how they did it was they took an egg cell and they took out the nucleus, and they took from a sheep, so they took from the mammary gland, I believe, one of the cells, and they took out the nucleus from the cell, and they put the nucleus into the egg. And so we've spoken a lot about biological age, and so this cell, the nucleus that was taken from the mammary cell, was taken from a sheep. I don't know what age the sheep was, but it wasn't, a biological age of 0. The sheep had aged. And so the question was, so the initial questions were trying to understand, as the cells differentiate and become different cell types, do the cells lose information about their original states? We now know that as cells replicate, they get the full copy of the DNA. It's not like half of it is replicated, and they only accumulate a portion of it. They have all the information capable of replicating and becoming different cell types, but the idea is that cell is differentiated and is now doing a specialized function. And so they wanted to know, if you took a specialized cell and took out the nucleus, which has the DNA sequence, and put it into an egg, would that egg then be able to create an organism that was a fully functioning organism, or would it just create a load of mammary cells? That was the question that they were trying to address. Like, does the cell lose information as it becomes specialized? And so what they found was that obviously you take the nucleus and put it into the egg, they were actually able to form Dolly, which was like, although she did die, I believe, somewhat shorter than they expected, she was otherwise functioning, didn't have any sort of abnormalities. And so that suggested that clearly the nucleus of that specialized cell had all of the information it needed to create another organism, but it just wasn't being expressed in the cell type. And so that information was being suppressed. And so this kind of also brings us into something known as cellular reprogramming. What are the triggers that causes a specialized cell to sort of differentiate and de-differentiate and go the other way and go back to a sort of stem cell that has the potential to form all of the different cell types that make up our body? And so that also would bring us into the entire field of regenerative medicine, and how potentially in the future we could take specialized cells like from our skin, which are easy, and we also are always making more skin cells, so it's easy to take and use. And we could then de-differentiate them back into a stem cell, and then we can sort of trigger that stem cell to turn into lots of different cell types. And then you could use that to create an organ for your heart, or use it to make neuron cells. I think this is all the different possibilities people are currently exploring. And so we might have a damaged organ as we age, and instead of depending on waiting for ages for a transplant, we could use our own body cells, create stem cells, and use them to make different organs. And that's the kind of premise behind regenerative medicine approaches. And so people are just trying to work out the safe ways or the effective ways of being able to do this in a controllable manner. So we did deviate a little bit from Dolly, but
no, but
I think
the
case of Dolly, Dolly sort of highlights what's possible. And obviously we don't want to be cloning humans at this point, but on a micro scale, we could be able to effectively somewhat clone an organ of ourselves and use it in regenerative medicine. And as opposed to either waiting for transplants. And then the other benefit is that it's our own cell type, so there isn't that host to graft mismatch. So there's not going to be some massive immune response to our own cells still. So super exciting work. I hope that we continue to see more advances with it.
Yeah, wonderful. So can you briefly explain to us Sinclair's work and his group's work on vision restoration by cellular reprogramming that you described in your video?
Exactly, yeah. So this kind of just leads on from what we just said. So cellular reprogramming is about taking a sort of specialized cell and converting it back to a stem cell, or now actually the hot topic is so-called partial reprogramming. So you don't fully go back to the stem cell, but you go back enough that the cell can start to replicate. And so in Sinclair's paper, they use some reprogramming factors. So these are factors that, when expressed in a cell, can trigger this dedifferentiation process that enables the cell to then replicate and then repair any damage. So what they did in mice is they sort of damaged the eye, and then they delivered these reprogramming factors, and effectively that enabled some of the cells to dedifferentiate and then replicate and replace. And you can actually see in their paper how it replaced in I think it was I forget exactly which part of the eye it was, but you could see the regrowth, and that regrowth was also associated with function, and it restored the vision in the mice that had been injured. And I believe they also did it in old mice, and it sort of were losing their vision, and it restored the vision as well. So the way they did that was with 3 of the reprogramming factors, OCT4, SOX2, KLF4, and they didn't use a protein called MYC, which is commonly upregulated in different cancers. So it was an interesting study, and I forget as well, I think they used a viral, so an AAV virus that can give the cells the different genes. So there is potential for human translations, because obviously these viruses are the sort of safer viruses that can deliver genes into the body. But again, I think we just have to wait for these things to be proven that they are safe. And then obviously there'll be a massive clinical trial going on. I'm not entirely sure how far, I'm not entirely sure if Sinclair's lab are even still studying this. I guess we'll have to wait and see. But yeah, I think that's just one example. Obviously, the eye is very accessible for things like delivery of these viruses. The question is, can we use the same technique and apply it for different tissues within the body? How can we get specificity to a certain tissue? But for sure, loads of people are really actually investigating this. There's tons of companies doing very similar things. So I'm pretty somewhat optimistic. For me being the biochemist, I just love to know how is this process happening? How do we go from a differentiated cell back to a stem cell? What can we learn about it? So yeah, super cool stuff.
Super futuristic. Thank you for sharing with us. I have a very quick last question for you, Eleanor. That is, what does rejuvenation mean, and what do rejuvenation biotech companies want to achieve? What are the most common strategies they try to employ to achieve what they propose? So rejuvenation strategies and possibly the role of blood factors in rejuvenation.
Cool. Yeah, I mean, for sure, that's not a quick question. But effectively, yeah, the way I think about the aging field or the longevity field in general is that you can sort of split the intervention approaches into 2 cohorts. One are more duroprotective strategies that might slow down the progression of aging, and others are these sort of rejuvenative approaches that kind of push back the aging process or they reverse it. So again, reverse, I don't know if that's entirely the right idea. I mean, I would include regenerative medicine in this category. So as you mentioned, with these different organs, replacing one organ with one you generated from a different cell in ex vivo and retransplanting that, I think would come under rejuvenation. But there's also a host of different other potential ways we could rejuvenate and actually reverse the process. And so again, reversing in this context would therefore potentially mean a reduction in the biological age, which again, we could then go, so it's kind of like a catch-22, how do we know these rejuvenative methods are working by using these biological aging measurements if we can't actually validate the biological aging measurements with known rejuvenation processes? But yeah, so I got a bit distracted. So one way you mentioned is with blood factors. So there's these classic experiments done called heterochronic parabiosis, where you can fuse the circulatory system of a young and an old mice, and you see that the old mice get rejuvenated. And so rejuvenation in this context was mainly looking at things like neurogenesis, so the growth of new neurons in the brain, as well as regrowth in different tissues within the body. And so subsequent work has been done from that, because obviously doing something like that is not something we want to be doing with humans. Neither do we want to really be harvesting young bloods and giving it to old people. So it turns out that actually, instead of finding young factors, if you simply dilute the blood of old mice, so you just take out the blood, get rid of everything but the blood cells, and put in just some saline solution back into the mice, that also showed similar rejuvenative effects. And so for me, that's something that seems quite promising. And so with these companies, majority of them that I've looked into are trying to use the cellular reprogramming strategy to basically work out what is the most effective way to generate stem cells, or to do this partial reprogramming to make it safer. Because obviously one concern with full reprogramming is you go back to a stem cell that's undifferentiated and potentially could turn into lots of different types of cells. Whereas with partial differentiation, the idea is that you regain cell proliferation, but the cell somewhat retains its identity. So if it was in the liver, for example, if it partially dedifferentiated, it could replicate, but it's also like, I know I'm a liver cell, and it would differentiate then into a liver cell, as opposed to becoming a neuron or something really strange. So yeah, I mean, obviously these companies are also somewhat secretive about what they're really up to, and so that's probably as much as I honestly do know. Obviously I know I did make a video on it and list some of the companies, and so they've probably updated their website since I last looked. But yeah, I mean, again, it's just, especially with Altos Labs, is probably the big headliner at the moment, with billions to create these different sensors for research into rejuvenation and what's causing aging, etc. So yeah, exciting times to be in the field, and so, yeah, I look forward to helping, at least to either communicate the science or even maybe to contribute to the science myself. So yeah.
Thank you so much, Eleanor. We've been lucky to hear about your extended knowledge on aging research, and I think we are also lucky to have someone like you that really keeps us updated on such a topic with, I mean, science precision, but at the same time in an engaging way. So thanks for the work you do with your channel, and I'm looking forward to seeing many more of your videos and maybe interviewing you again someday. So thank you so much for the time you spent with me.
And also, thank you for having me. It's always fun to communicate science.
You've just listened to About a Futurist, a podcast by Luca Fusar-Bassini. This is the first series, and a new episode is out every Monday. Please consider subscribing and rating the podcast on Spotify, Apple Podcast, YouTube, Instagram, or your top podcasting platform. And if you liked this episode, consider sharing it with your friends, as the growth of new podcasts relies on word of mouth. If you have any suggestions, don't hesitate to reach out to me on Instagram or Gmail at thebiotechfuturistpodcast@gmail.com. You can find the full AI-generated transcript of this episode on my website, lucafusarbassini.com. I'll also post the links to the main papers referenced in this episode, which you can find here in the description too. Thanks for listening to A Biotech Futurist. I am looking forward to talking with you in a week.